start our next unit 1.2, the ultrastructure of cells. So here we have some understandings. Prokaryotes have a simple cell structure without compartmentalization. Eukaryotes have compartmentalized cell structure, and electron microscope have a much higher magnification than light microscope. So here are some application and skills. If you want, you can do it. And now, so what is a prokaryotic cell? After extensive studies of cells, it has become apparent that all cells use some common molecular mechanism. There are huge differences between different forms of life, but cells are the basic unit, and different cells have many characteristics in columns. Cells are often divided into particular groups based on the major characteristics. One such division separates cells into two groups, prokaryotic and eukaryotic cell. Prokaryotic cells are much smaller and simpler than eukaryotic cells. In fact, most prokaryotic cells are less than 1 millimeter, like the micrometer in diameter. So prokaryotic cells are much smaller and simpler than eukaryotic cells, that we need to remember, and there are huge differences between the forms of life. So because of this and many other reasons that will be discussed later, the prokaryotic cells are thought to have appeared on the earth first. As the bacteria are prokaryotic cells, you can see that such cells play a large role in the world today. Now here we have some features of the prokaryotic cells. Study the figure of the prokaryotic cell in figure and make sure you can identify the cell wall, plasma membrane, flagella, pili, ribosomes, and the nucleoid region containing free DNA. Here we have some information. Bacteria and members of groups referred to as archaea are made up of prokaryotic cells and are called prokaryotes. The vast majority of these organisms do not cause diseases and are non-pathogenic disease causing. Okay, so that's good. Now here we have the diagram. Here this is the this is the bacteria cells. Yeah, these are bacteria cells, and this is a figure shown, and has been labeled with the names of the parts. And the outer layer, it is like the capsule, and the inner, this yellow part, is the cytoplasm. Ribosomes are the structure, and cell wall is the cell wall surrounding it, and the plasma membrane and nucleoid of the DNA, plasmids, pili, and flagella. So the cell wall and plasma membrane, the prokaryotic cell walls protects the and maintains the shape of the cell. So the prokaryotic cell wall protects and maintains the shape of the cell. In most prokaryotic cells, this wall is composed of carbohydrate protein complex called peptidoglycan. Some bacteria have an additional layer of a type of polysaccharide outside the cell wall. This layer makes it possible for some bacteria to adhere to structures such as teeth, skin, and food. So some bacteria have additional layer of the type of polysaccharide outside the cell wall in order to maintain its shape, and this layer makes it possible for bacteria to adhere to structures such as teeth, skin, and food. Now, the plasma membrane is found just inside the cell wall and is similar in composition to the membranes of the eukaryotic cell. So, plasma membrane is just inside the cell wall, and to a large extent, the plasma membrane controls the movement of the materials into and out of the cell, and it plays a role in the binary fission of the prokaryotic cell. So, it plays a major role in the binary fission. The cytoplasm occupies the complete interior of the cell. The most visible structure with the microscope capable of the high magnification is the chromosome or the molecule of DNA. There is no compartmentalization within the cytoplasm because there are no internal membranes other than the plasma membrane. And so therefore all cellular processes within prokaryotic cell occur within the cytoplasm. So all processes occur in the cytoplasm only. And pili and flagella. Some bacterial cells contain hair-like growths on the outside of the cell wall like this. These structures are called pili and can be used for attachment. However, their main function is joining bacterial cells. So the main function of these two are to join the bacterial cell in the preparation for the transfer of DNA from one cell to another. And some bacteria have flagella, plural. This is the plural form, like many, and flagellum, it's singular, which are longer than pili. Flagella allows a cell to move, so the main function is also like to move. Okay, This is a false color transmission electron micrograph TEM showing E. coli dividing by binary fission. Okay, so this is how the cells divide and if seen under microscope. So ribosomes occur in all prokaryotic cells and they function as site of the protein synthesis. Ribosomes are present in all prokaryotic cells and so they function as the site of protein synthesis. They make up proteins there. These small structures occur in very large number in the cell that produces a lot of proteins and when numerous they give a granular appearance to electron micrograph of the prokaryotic cell. And the nucleoid region of the bacterial cell is the non-compartmentalized and contains a single long continuous circular thread of DNA, the bacterial chromosomes. So nucleoid region of the bacterial cell is non-compartmentalized. It is not compartmented uh, into different parts and contains a long single continuous circular thread of DNA, the bacterial chromosomes. Therefore, this region involved with the cell control and reproduction. In addition to the bacterial chromosomes, bacteria may also contain plasmids. They have plasmids. These small circular DNA molecules are connected to the main bacterial chromosome. The plasmids replicate independently of the chromosomal DNA, and plasmid DNA is not required by the cell under normal condition, but it may help the cell adapt to unusual circumstances. And now here is some information. The importance of the plasmids in the prokaryotic cells will be discussed fully in chapter 3. Plasmids have very important roles to play in some techniques involving genetic engineering and modification. So plasmids have a very important role to play. Okay, now binary fission. Prokaryotic cells divide by a very simple process called as binary fission. So this is a process of reproduction, like they divide and increase their population. During this process, the DNA is copied. The two daughter chromosomes become attached to the different regions on the plasma membrane and the cell divides into the two genetically identical daughter cells. This divisional process includes an elongation of the cell and a partitioning of the newly produced DNA by microtubule-like fibers called FTS. Okay, so a summary. Here is the list of major distinguishing characteristics of the prokaryotic cells. Their DNA is not enclosed within the membrane and forms one circular chromosome. And DNA is free. It is not attached to proteins, so DNA is completely free. They lack membrane-bound organelles, and ribosomes are complex structures within the plasma membrane, but they have no exterior membrane. And their cell wall is also made up of compound called peptidoglycan. They usually divide by binary fission. A simple form of this is a whole the summary we are reading and form of cell division. They are characteristically small in size, usually between 1 to 10 micrometers. And now, what is a eukaryotic cell? Whereas prokaryotic cells occur in bacteria, eukaryotic cells occur in organisms such as algae, protozoa, fungi, uh, plants, and animals. And now, examining the following diagrams and pictures. Okay, so this is a eukaryotic cell. Here we have the nucleus and it is the nuclear membrane surrounding it. Here we have nucleolus, which is a dense solid structure involved in the ribosome synthesis. And now here we have Golgi apparatus here also, which stores and modifies and packages proteins, and nuclear pores here allows the communication between the nucleus and the rest of the cell. And ribosomes, the function of ribosome are small. They are the small structure and free in the cytoplasm or associated with the endoplasmic reticulum (ER). And now here is the rough endoplasmic reticulum is a site of protein synthesis where the synthesis of protein takes place, where protein is made up. Now smooth endoplasmic reticulum is the ER without ribosomes. Vacuoles are smaller than those found in the plants. Vacuoles here, these blue regions are the vacuoles and are smaller than those found in the plants. And lysosomes, these green color are the sacs bounded by a single membrane. They contain and transport enzymes. Lysosomes show little internal structure. Lysosomes are usually absent from the plant cells. And here we have centrioles, which are associated with the nuclear division. They are composed of the microtubules, the area in which the centrioles are found in is called the centrosome. It is present in all eukaryotic cells, but centrioles are absent from higher plant cells. And mitochondria cytoplasm is this whole yellow structure. And mitochondria are organelles that carry out respiration. So their function is to carry out respiration. Now, eukaryotic cells range in the diameter from 5 to 100 micrometers. A kernel or a nucleus is usually noticeable in the cytoplasm. Other organelles may be visible within the cells if you have a microscope with high enough magnification and resolution. Organelles are non-cellular structures that carry out specific functions, a bit like organs in the multicellular organisms. Different types of cells often have different organelles. These structures enable compartmentalization in eukaryotic cells. So they help in compartmentalization in eukaryotic cells, which is not a characteristic of prokaryotic cells. They do not have this feature. So compartmentalization enables different chemical reaction to be separated, which is especially important when adjacent chemical reactions are incompatible. And compartmentalization also allows chemicals for specific reactions to be isolated. This isolation results in a increased efficiency. So in a sense, it's good. The term eukaryotic comes from the word eukaryon. It's a Greek word meaning true kernel or the true nucleus. Here is a structure. We have another plant cell. So this here blue structure we have the central vacuole has storage and hydrolytic functions. And here we have endoplasmic reticulum. ER is a network of tubes and flattened sacs. ER connects with the plasma membrane and the nuclear membrane and may be smooth or have attached ribosomes. Now, cytoplasm contains dissolved substances, enzymes, and the cell organelles. Here, the cytoplasm, the empty space here. Nucleus contains most of the cell's DNA, and the nuclear pore here, and the nucleus, and the nuclear membrane is a double-layered structure. Ribosomes are 20 nanometer structures that manufacture proteins. They may be free in cytoplasm or associated with the surface of the endoplasmic reticulum. Golgi apparatus here, and starch granules are composed of carbohydrates stored in amyloplast, and mitochondria are bounded by a double membrane. They are energy transformers. Plasma membranes, they are found, it is inside, just inside the cell wall. Cell wall is a semi-rigid structure composed mainly of the cellulose. It is made up of cellulose. Now, chloroplasts are specialized plastids containing the green pigment chlorophyll. So here is the place where we have chlorophyll. They consist of the grana within the colorless stroma. They are the sites for protein for photosynthesis. So it is the microscopic picture that we can see here of the pancreatic exocrine cell. Can you tell this is it an animal cell? Locate as many structures of an animal cell you can. And now, how do the structure of this cell reflect overall function of the pancreas? Okay. Now, here we have another a TEM of the cell from the mesophyll region of a leaf. How do you know this is a plant cell? Okay, so you have to answer. Locate as many of the structures of the plant cell as you can. What cell structures reflect most the unique abilities of the plant cell? As you read about the organelles of the eukaryotic cell below, refer back to the figures above the page 16 and add more names to the organelles. Also, be certain to note which organelles are common to both type of cells and which occur in only one of the two types. Organelles of the eukaryotic cells, which are endoplasmic reticulum, ribosomes, lysosomes, which are not usually found in plants, Golgi apparatus, mitochondria, nucleus, chloroplast only are found in plant and algal cells, and centrosomes in all eukaryotic cells, but centrioles are not found in some plant cells and vacuoles. The microscope has given us an insight into the structure and the function of the following eukaryotic cells, organelles, and characteristics. Now, cytoplasm, all eukaryotic cells have a region called cytoplasm that occurs inside the plasma. membrane or the outer boundary of the cell. It is in this region that the organelles are found. These are found only in cytoplasm, and the fluid portion of the cytoplasm around the organelle is called the cytosol. So the fluid portion is known as cytosol. Endoplasmic reticulum (ER) is an extensive network of the tubules or the channels that extend most everywhere in the cell, from nucleus to the plasma membrane. Its structure enables its function, which is transportation of the materials throughout the internal region of the cell. So its function is to for the transportation of the materials throughout the internal region of the cell. There are two general type of ER: endoplasmic reticulum, smooth endoplasmic reticulum, and rough endoplasmic reticulum. Smooth ER does not have any of the organelles called ribosomes or on its exterior surface. So the smooth endoplasmic reticulum, they do not have ribosomes on its exterior surface like the outer surface. Rough endoplasmic reticulum has ribosomes on its exterior surface. Now here we have a diagram of the smooth endoplasmic reticulum and the rough endoplasmic reticulum. So this one is the smooth, and rough endoplasmic, it has the ribosomes out here that we can see, and it is the nuclear membrane, and this is the nucleus here. So the smooth endoplasmic reticulum has many unique enzymes embedded on their surface. Its function are the production of membrane, phospholipids, and cellular lipids, the production of the cell hormones such as testosterone and estrogen, detoxification of the drugs in the liver, storage of calcium ions in the muscle cells needed for the contraction of muscle cells, and the transportation of lipid-based compounds, helping the liver release glucose into the bloodstream when needed. Rough endoplasmic reticulum has ribosomes on the exterior of the channels. These ribosomes are involved in protein synthesis. Therefore, this type of AR is involved in the protein development and transport. These proteins may become part of membranes, enzymes, or even messenger between the cells. Most cells contain both type of ER, with the rough endoplasmic reticulum being closer to the nuclear membrane. Now, ribosomes are what are ribosomes? Ribosomes are the unique structures that do not have an exterior membrane. They carry out protein synthesis within the cell. Ribosomes carry out the protein synthesis within the cell where protein is made up. These structures may be found free in the cytoplasm, or they may be attached to the surface of the ER. They are always composed of a type of RNA and protein. You will recall the prokaryotic cells also contain ribosomes. However, the ribosomes of the eukaryotic cells are larger and denser than those of the prokaryotic cells. Ribosomes are composed of the two subunits. These subunits together equals 80s. The ribosomes in the prokaryotic cells are composed of the two subunits, but they only equal to 70s. Lysosomes. Lysosomes are intracellular digestive centers that arise from the Golgi apparatus. A lysosome does not have any internal structure. Lysosomes are sac-bounded by a single membrane that contain as many 40 different enzymes. The enzymes are all hydrolytic and they catalyze the breakdown of the protein, nucleic acid, lipids, and the carbohydrates. Lysosome fuse with the old or damaged organs from within the cell to break them down so that recycling of the components can occur. Lysosomes are also involved in the breakdown of materials that may be brought into a cell by phagocytosis. Phagocytosis is a type of endocytosis that is explained on page 36 in the section 1.4 that we will read. And the interior environment of the functional lysosome is acidic. So the interior lysosome is acidic. This acidic environment is necessary for enzymes to hydrolyze large molecules. Now, Golgi apparatus. The Golgi apparatus consists of what appears to be flattened sacs called cristae, which are stacked one top of the other. This organelle functions in the collection, packaging, modification, and distribution of materials synthesized in the cell. So the function is to modify, distribute materials, and package and transport in the cell. One side of the apparatus is needed, the rough endoplasmic reticulum, called the cis side, and it receives products from the ER endoplasmic reticulum. These products then move into the cristae of the Golgi apparatus. They continue to move in discharging or the opposite side, the trans side, which you call the trans side. Small sacs called vesicles can then be seen coming off the trans side. These vesicles carry modified materials to wherever they are needed, inside or outside the cell. These organelles are especially prevalent in the glandular cells, such as those in the pancreas, which manufacture the secrete substances. Here is the diagram that you can see. Now mitochondria, singular mitochondrion. When one we call it mitochondrion, and when they are many, they are mitochondria. Are rod-shaped organisms that appear throughout the cytoplasm. They are present in cytoplasm. They are close in size to the bacterial cell. Mitochondria have their own DNA, a circular chromosome similar to that of the bacterial cell, allowing them some independence within a cell. They have a double membrane. The outer membrane is smooth, but the inner membrane is folded into cristae. As you can see in the diagram, the singular crista. Inside the inner membrane is a semi-fluid called the matrix. We call it matrix, and area called the inner membrane. Space lies between the two membranes. The cristae provide the huge surface area within the chemical reaction characteristic of the mitochondria occur. Most mitochondrial reactions involve the production of the usual cellular energy called the adenosine triphosphate ATP. Energy is stored in this form. Because of this, the mitochondria are often called the powerhouse of the cell. And the organelle also produces and contain its own ribosomes. Their ribosomes are of the 70s type. Uh, Cells that have high energy requirements, such as the muscle cells, have large number of mitochondria. So the muscle cells have large number of mitochondria. They need more energy. Here is the matrix, and these are the cristae, inner membrane, outer membrane. Here, as you can see, and also in this microscopic figure. Now, nucleus in the eukaryotic cell is an isolated region where DNA resides, where we found DNA. It is bordered by double membrane referred to as the nuclear envelope. This membrane also allows compartmentalization. So this allows compartmentalization of the eukaryotic DNA, thus providing an area for the DNA to carry out its function without being affected by the process occurring in the other parts of the cell. The nuclear membrane does not provide complete isolation because its numerous pores that allow communication within the cell cytoplasm. Here we have another figures, some of the figures. DNA of the eukaryotic cell often occurs in the form of chromosomes. Chromosomes vary in the number depending on the species. Chromosomes carry all the information that is necessary for the cells to exist. This allows an organism to survive, whether it is unicellular or multicellular. The DNA is genetic material of the cell. DNA is the genetic material of the cell. It enables certain traits to be passed on to the next generation when the cell is not in process. So here we have a chromosome, and it is the DNA wrapped around the histone core, held in the place with another histone, and central core of the nucleosomes made up of 8 histones. Now, when the cell is not in the process of dividing, the chromosomes are not present as the visible structure. During this phase, the cell's DNA in the form of chromatin, and the chromatin is formed of the strands of the DNA and proteins called the histones. The DNA and histone combination often results in the structure called the nucleosome. The nucleosome consists of the 8 spherical histones with a strand of DNA wrapped around them and secured with the ninth histone. This produces a structure that resembles a string of beads. A chromosome is highly coiled structure of many nucleosomes. The nucleus is often located centrally within the cell cytoplasm, although in some cell types it is pushed to one side or the other. The side position is the characteristic of the plant cells because these cells often have a large central vacuole. Most eukaryotic cells possess a single nucleus, but some do not have a nucleus at all, and some have multiple nuclei without a nucleus. Cells cannot reproduce. The loss of productive ability is often paired with the increased specialization to carry out the certain function. For example, a human red blood cell do not have nuclei. They are specialized to transport respiratory gases. Most nuclei also include one or the more dark areas called the nucleoli. Ribosomes molecules are manufactured in the nucleolus. The molecules pass through the nuclear envelope before assembling as ribosomes. And now here we have chloroplast. Chloroplast occur only in algae and the plant cells. Human beings do not have chloroplast, and animals do not have chloroplast. Birds also don't have chloroplast. Fish also don't have chloroplast, and chloroplast contains a double membrane and is only the same size as the bacterial cell, like the mitochondria. The chloroplast contain its DNA and the 70s ribosome. The DNA of the chloroplast takes the form of a ring. You should note that all the characteristics that chloroplast and the mitochondria have in common with the prokaryotic cell, as well as the DNA and the ribosome. The interior of the chloroplast includes the grana, singular granum, and thylakoids and the stroma, which are labeled in the figure. Granum is made up of the numerous thylakoids stacked like a pile of coins. The thylakoids are flattened membrane sacs with the components necessary for the absorption of light. Absorption of light is the first step in the process of photosynthesis. The fluid stroma is similar to the cytosol of the cell. It occurs outside the grana, but within the double membrane. Stroma contains many enzymes and chemicals that are necessary to complete the process of photosynthesis. Like the mitochondria, chloroplasts are capable of reproducing independently of a cell. So, just like mitochondria reproduces, chloroplasts can also reproduce and divide more easily. Here we have a structure of chloroplast. This is the chroma, and this is one thylakoid shown here, and outer membrane, inner membrane, and granum, the pile of thylakoids. So here we have the centrosome. The centrosome occurs in all the eukaryotic cells. Generally, it consists of the pair of the centrioles at the right angle to one another. These centrioles are involved with the assembly of the microtubules, which are important to cell because they provide structure and allow movement. The microtubules are also important for the cell division. Cells from higher plants, plants that are thought to have evolved later, produces microtubules even though they do not have centrioles. Okay, so the plants that came to exist came to existing life earlier, they don't have much functions as the plants that came after. Okay, so the centrosome is located at the end of the cell close to the nucleus. Vacuoles. Vacuoles are the storage granules that are usually formed from the Golgi apparatus. They are the membrane bound and have many possible functions. They occupy a very large space inside the cell of the most plants. They may store a number of different substances, including potential food, metabolic waste, toxins, and water. Okay, so they also store number of different substances, including potential food, like to provide nutrition, and metabolic waste, toxins, and water also. So vacuole enables cells to have higher surface area to volume ratios even at larger sizes. Okay, and in plants they only allow the uptake of water, so they only help to store the water maybe, which provides rigidity to the organism in good shape and good life. So here we have some differences comparing prokaryotic and eukaryotic cells. DNA in the ring. DNA. Okay, prokaryotic cells have DNA in the ring form without protein, and eukaryotics have the DNA with protein as chromosome or chromatin. Now DNA free in cytoplasm, and here we have DNA enclosed with the nuclear envelope nucleus. And prokaryotic cells have no mitochondria; they have mitochondria present, and prokaryotic cells have 70s ribosomes and they have 80s ribosomes. Prokaryotic cells have no internal compartmentalization to form organelles, and they are less, and their size is less than 10 micrometers. And here in eukaryotic cells, we have internal compartmentalization present to form many types of organelles, and the size is more than 10 micrometer. If asked to state the similarities between the two cells, make sure you include the following points. Okay, so let's read. Both type of cells have some sort of outside boundary. That always involves a plasma membrane. Okay, good point. Both types of cells carry out all the function of life. DNA is present in both type of cells. Okay. Now here we have a table showing the comparing the plant and the animal cells. Okay. Let's read. The exterior of the cell includes an outer cell wall and with a plasma membrane just inside. But animal cell do not have a cell wall; they just have a plasma membrane. Chloroplasts are present in the cytoplasm area, but in animal cell there are no chloroplasts because photosynthesis does not take place in animal cells. Large centrally located vacuoles are present in plant cells, but in animal cells, vacuoles are not usually present or are small. Carbohydrates are stored as starch in plant cells, but carbohydrates are stored as glycogen in animal cells. Plant cells do not contain centrioles within a centrosome area, but animal cells contain centrioles within a centrosome area. Because a rigid cell wall is present, this cell type has fixed, often angular shape. But animal cells without a cell wall, the cell is flexible and more likely to be a rounded shape. Plant cells have more advantage or benefit because of cell wall. They can be in fixed shape. Most cell organelles are present in both plant and animal cells. When an organelle is present in both type of cells, it is usually the same structural function. For example, the cell types contain mitochondria that possesses cristae, a matrix, and the double membrane. Also, in the both type of cells, the mitochondria function in the production of ATP for the use by the energy by the cell. Energy used by the cell. Now here we have the outermost parts of the different cells. Bacteria, cell wall of peptidoglycan. Fungi has cell wall of chitin. Yeast has cell wall of glucan and mannan. Algae has cell wall of cellulose. Plants have cell wall of cellulose. Animals have cell wall, no cell wall. Instead of plasma membrane, they secrete a mixture of sugar and protein called glycoproteins that form the extracellular matrix. Quite interesting. They produce the mixture of sugar and proteins called the glycoproteins that form the extracellular matrix. Here we have a diagram. Here is the cells. Here we have the cells. This is the middle lamella. We have the secondary wall and the primary wall here. So whenever cell is present, it is involved in maintaining the cell shape. It also helps to regulate the water uptake because the rigidity. It also helps in maintaining the water level. Okay, so it's good. Because of the rigidity, it will only allow a certain amount of water to enter the cell. In plants, when an adequate amount of water is inside the cell, there is pressure against the cell wall. This pressure helps support the plant vertically. Okay. The extracellular matrix ECM of many animal cells is composed of the collagen fibers plus the combination of the sugars and the proteins called the glycoproteins. These form fibers like structures that anchor the matrix to the plasma membrane. Okay, hope you understand. This strengthens the plasma membrane and allow attachments between the adjacent cells. The ECM allows the extracellular matrix allows cell-to-cell interactions, possibly altering gene expression and enabling the coordination of the cell actions without the tissue. Many researchers think the extracellular matrix is involved in the directing stem cells to differentiate. Cell migration movement also appear to be at least partially the result of interaction in this area. Here is the diagram. Here we have the collagen fiber and the glycoprotein, which are the ECM, and integral proteins in the plasma membrane and the microfilaments. Okay, this is the cytoplasm. Here we have some activity. If you have time, you can do that. And here are some exercises. Okay, so that's all for today. Thank you.